All right, so today we're diving into B cell receptors, or BCRs. They're like these tiny gatekeepers on our B cells. And we want to understand, how they actually detect this crazy variety of invaders and kickstart our immune response.
It's pretty wild when you think about it.
Yeah, it really is.
I mean, we take our immune systems for granted,
Yeah.
but when you look at it, amazing how complex it is.
Yeah, before we jump into that too much, you shared some great sources with us that really outline, how BCRs are assembled and how they work. So where do we even begin?
Well, to understand BCRs, you got to think about immunoglobulins or IgGs, or as most people know them, antibodies.
Right, the Y-shaped proteins we hear about all the time.
Yeah, like the guided missiles of the immune system.
Exactly.
And a BCR is basically a membrane-bound immunoglobulin, so it's stuck right there on the surface of the B cell.
So it's like an antibody that's attached to the B cell just waiting there.
Exactly. And each BCR is made up of 4 protein chains, 2 identical heavy chains, and 2 identical light chains. The heavy chains are what anchor the BCR to the B cell's membrane, and then the light chains, along with parts of those heavy chains, they actually form what we call the antigen-binding site.
So that antigen-binding site, that's what makes each BCR so specific, right?
Yeah.
It can recognize a very specific target.
Exactly. And what's really mind-blowing is the sheer diversity of those antigen-binding sites.
Yeah.
Our bodies can generate BCRs for almost any antigen that we might run into.
So you're saying our bodies can make antibodies for practically anything?
Yeah.
I mean, how is that even possible?
It's through this process we call gene rearrangement and somatic hypermutation. So basically during B cell development,
Okay.
the genes that code for those heavy and light chains we were talking about, they go through this sort of shuffling process and mutation.
Okay.
It's kind of like you're taking a deck of cards and just constantly shuffling them, creating millions of different combinations.
Oh, so it's not like there's one gene for every single antigen out there.
No, it's way more elegant than that.
Yeah.
This gene rearrangement process allows us to take just a small number of genes and make a huge repertoire of unique BCRs.
That's incredible. It's like this master key that you can adjust to fit any lock.
Yeah, and on top of that, you have somatic hypermutation, where those genes encoding the antigen binding site, they can actually undergo even further mutations to really fine-tune the BCR's specificity.
Wow. Okay, so we've got this incredible system for creating all these different BCRs, each one with its own special target. But what happens when a BCR actually bumps into its matching antigen?
That's when things get really exciting.
Okay.
When that antigen binds to the BCR, it sets off this chain reaction of signals inside the B cell.
It's like a signal flare going up.
Right.
Okay, there's an intruder here.
Right. You don't want just one guard on the city wall to, launch a full-scale attack every time they see something a little suspicious.
Right. You need to check it out first.
You need a communication system. You need coordination.
So the BCR is not just like an on-off switch. It's more like sending a report back to headquarters.
Yeah, like, hey, I found something. What do we do?
Yeah.
And that signaling cascade ultimately leads to something we call B cell activation.
And what does that mean? What's happening when a B cell is activated?
So it involves this process called clonal expansion, where that activated B cell just starts dividing like crazy, creating a whole army of clones.
Yeah.
And each clone has the same BCR, all targeting that one specific antigen.
It's like this super specialized task force.
Yeah, exactly.
That goes in to fight the invasion.
Exactly.
But I'm guessing it doesn't just end there.
You're right. There's another layer we have to talk about.
Okay.
Class switching.
Class switching, what's that?
So imagine you have this army of B cells, right?
Yeah.
They all have the same BCRs. They're targeting that antigen.
Right.
But they need to be able to fight this invader in different parts of the body and use different tactics. That's where class switching comes in.
Okay.
It's like adapting those troops for different battlefields.
I like that analogy.
It happens in these specialized areas within lymph nodes called germinal centers.
Okay.
During class, switching the heavy chain of that BCR, it actually gets modified.
Oh.
And it results in different antibody isotypes.
Like IgM and IgD?
You got it.
IgA?
Each one has a specific function.
Right. Like IgM is like that first responder, and then IgG provides longer-term immunity.
Exactly. Each isotype has unique properties that make it better suited for certain tasks.
Yeah.
And this adaptability, that's what makes for a truly effective immune response.
It's just amazing how the immune system can be so precise in its response.
Yeah.
you know?
Yeah.
We've got this incredible diversity of BCRs, the antigen binding, the signaling, and then this adaptation through class switching. And it's so cool how it happens step by step.
It's a hallmark of humoral immunity.
Right.
And it's important to remember that this whole system of BCRs and antibodies is just one piece of the puzzle when we talk about our immune response.
Yeah, it's all connected.
Yeah.
But before we get too ahead of ourselves, let's pause here.
That's a good idea.
Give our listener a moment to process all this.
Yeah.
We'll be back in just a minute to dive even deeper into the world of BCRs and how they keep us healthy.
Welcome back. I hope you've had some time to think about, BCR assembly and activation. What's sticking out to you so far?
Honestly, it's a lot to take in millions of unique BCRs.
Right.
Each one able to recognize a specific antigen. And it all comes from just a small number of genes.
Yeah, it's really a remarkable feat of biological engineering.
It really is, and it shows how powerful evolution is, to create this adaptable and efficient system of defense.
Yeah, totally.
So we talked about how when an antigen binds to a BCR, it triggers that cascade of signaling events inside the B cell. But you said something earlier that really piqued my interest. You mentioned that BCRs aren't just simple on-off switches. What did you mean by that?
Well, remember when we talked about cyclic AMP signaling?
Yeah.
And those A kinase anchoring proteins, or AKAPs?
Vaguely, yeah. Those are the proteins that act as scaffolds, right?
Yeah.
Bringing together different signaling molecules.
Exactly. And they actually play a role in BCR signaling as well. They help organize and fine-tune the cellular response,
Okay.
making sure that the signal is transmitted effectively.
So it's not just about the antigen binding to the BCR, it's also about where the BCR is located and what's around it.
Yeah, exactly. It's all about context. The same signal can have totally different effects depending on that cellular environment.
Right.
And speaking of context, we also have to think about the overall health and state of the immune system.
Yeah, because a weakened immune system wouldn't be as good at fighting off infection, even with a perfectly functional BCR system.
Right. Absolutely. Things like stress age, underlying health conditions,
Yeah.
they can all impact immune function, including how BCRs respond to threats.
That makes sense. So even though BCRs are these incredibly sophisticated sensors, they're still part of this larger interconnected system.
Right.
And that brings us back to that question you asked earlier. If BCRs are so effective, why do we still get sick?
Yeah, the million-dollar question, it's a good reminder that biology is rarely simple. Even with a system as elegant and powerful as BCRs, there are always challenges.
Like what makes us susceptible to illness even with a strong immune system?
Well, for one, pathogens are constantly evolving.
Yeah.
They're always finding new ways to avoid our immune defenses.
Oh, so they're getting sneakier.
Yeah, like some viruses, they're masters of disguise.
Right.
Constantly mutating their surface proteins so that BCRs can't recognize them.
It's like an arms race.
Exactly, our immune system versus the pathogens.
Right.
And sometimes those pathogens win, at least for a little while. That's why we get sick.
And what about things like autoimmune disorders?
That's another layer of complexity in autoimmune diseases. The immune system actually attacks the body's own cells.
Wow, so it's like the immune system is turned against us.
Yeah, it's like it's gone rogue, mistaking friend for foe.
So in those cases, it's not that the BCRs aren't working, it's that they're targeting the wrong things.
Exactly. It's a case of mistaken identity with some pretty serious consequences. And then, of course, you have those external factors like exposure to toxins or pollutants,
Right.
things that can weaken our immune defenses.
So it's like this whole web of interconnected factors that determine whether or not we get sick.
It really is, and that's why it's so important to take a holistic approach to our health. Supporting our immune system through things like good nutrition, exercise,
Right.
stress management. It can make a huge difference.
So it's not just about relying on our BCRs to do all the work.
Right.
We have to create an environment where our immune system can thrive.
Exactly. And speaking of the bigger picture, let's go back to those comparisons to other receptor systems in the body. We touched on this a bit earlier,
Yeah.
but I think it's worth exploring a little more.
Yeah, you're right. We talked about how BCRs are similar to other receptors.
Right.
They bind to specific ligands and trigger that signaling cascade.
Yeah.
But what about the differences?
Yeah.
What makes BCRs unique?
Well, one key difference is that incredible diversity we talked about. The ability of BCRs to create millions, even billions of different antigen binding sites just by rearranging those genes.
Right.
It's really remarkable. Other receptor systems, they don't have that same level of adaptability.
It's hard to imagine how that much variation can come from shuffling genes around.
It really is mind-boggling. It's a testament to the power of evolution to create such a sophisticated system.
Yeah.
Another unique thing about BCRs is that class switching.
Right. Do other receptor systems do anything like that?
Not really. While other receptors might have different isoforms or subtypes,
Yeah.
they don't change their fundamental structure in response to a stimulus like BCRs do during class switching.
So that's another thing that sets BCRs apart.
Definitely, it's a brilliant strategy for tailoring that immune response to different types of pathogens and different locations in the body.
Okay, so we've got diversity and adaptability.
Yeah.
What else, what other insights can we get from these comparisons?
Well, it's interesting to think about the idea of dominant negative activity. Remember when we talked about thyroid hormone receptors and that NCORI protein?
Right. That's where like a mutant or altered protein can mess with the function of its normal counterpart.
Exactly. It's like having a saboteur inside your own cells.
That's kind of scary.
Yeah.
It makes you realize how fragile the balance is within these biological systems.
It does, and it makes you wonder if something similar could happen with BCRs.
Oh.
What if a mutation created a BCR that actually interfered with the normal immune response?
That's a fascinating thought, although a bit unsettling.
Yeah,
It really highlights how important research is in these areas.
right.
The more we understand about these processes, the better we'll be able to address any potential problems.
I agree, and it really underscores the fact that we're still just scratching the surface when it comes to understanding the immune system.
My brain is full. We've covered so much in this deep dive into B-cell receptors.
I know, right?
Let's take another quick break to let all this sink in, and then we'll come back to wrap up our exploration of these amazing molecular gatekeepers. All right, we're back and ready to wrap up our deep dive into these fascinating B-cell receptors.
Yeah, it's amazing how something so small can have such a big impact on our health.
I know, right? And we've been talking about all this intricate stuff, gene shuffling, antigen binding, cellular signaling.
Yeah, it's really mind-blowing.
It is just really shows how powerful evolution is, to create such a sophisticated system of defense.
Absolutely, and yet we still get sick.
I know.
It's humbling, right?
Yeah.
It makes you realize that even with all our scientific advances, we still have so much to learn about the immune system.
For sure. And that question, why do we still get sick? I feel like that's what makes immunology so fascinating.
It really is. It's this constant back and forth between our defenses and those ever-evolving pathogens.
Yeah, like a never-ending game of cat and mouse.
Exactly. Our BCRs and antibodies are trying to keep up with all the tricks that viruses and bacteria use.
It's like they're always one step ahead.
Right, and that's why research in this field is so important. The more we understand about BCRs, how they work, how they evolve, how we can maybe even manipulate them,
Yeah.
the better equipped we'll be to develop new therapies and treatments for all kinds of diseases.
As we've been talking about BCRs, I'm thinking about all the other things happening in our bodies. We talked about how BCRs are kind of like other receptor systems.
Right.
But are there any things that make BCRs really stand out?
Yeah, a couple things come to mind for me.
Okay.
One is that incredible diversity we talked about.
Right.
The fact that BCRs can create billions of different antigen-binding sites just by rearranging their genes, it's pretty amazing.
Yeah, and that's what allows our immune system to recognize and respond to so many different threats.
Exactly. It's like having a security system that can identify every single person on the planet, even people it's never seen before.
Wow, that's a great analogy.
And then there's that class switching we talked about, which allows those B cells to fine-tune their response, tailoring it to the specific type of pathogen and where it is in the body.
Yeah, like having a whole arsenal of different weapons.
Exactly. Each one designed for a specific job. And the more we learn about how class switching works,
Yeah.
the better we might be able to develop even more targeted therapies.
It's mind-blowing to think about all of this happening inside us right now at this microscopic level.
I know, it's incredible.
These tiny little machines are working so hard to keep us healthy.
It's truly a marvel of nature, and as we wrap up this deep dive, I think it's important to remember that our understanding of BCRs and the immune system is always evolving.
Right, there's always more to discover.
Exactly, and that's what makes science so exciting.
Yeah, it's a never-ending journey.
It is.
Well, I feel so inspired after this deep dive,
Me too.
It's given me a whole new appreciation for how complex and elegant the immune system really is.
I hope our listeners are feeling the same way.
As we're wrapping up, I keep thinking back to that big question. If BCRs are so effective, why do we still get sick?
It's the question that keeps us curious.
Yeah.
Keeps us pushing forward, trying to understand more.
Yeah, and even though we don't have all the answers yet, every new discovery gets us closer to understanding this amazing system that keeps us alive.
That's the beauty of it.
Well, thank you for joining us on this incredible journey into the world of B-cell receptors. We hope you'll tune in again for another deep dive into the world of science and discovery.